start. I will take the attendance at the end of the session, inshallah. So today we are going to start a new topic, which is enzymes, chapter 5 from your textbook. All right. So please open page number 49 from your textbook. We are going to start enzymes today. So let's start with the verse from the Quran, which is related to enzymes. Okay, so this is a verse from chapter 16, verse 68 and 69, according to which Allah says, your Lord inspired to the beasts, saying, take your habitations, meaning make hives in the mountains and in the trees and in what they build. Okay, and then eat from all the produce of the earth and follow the spacious paths of your Lord made easy for you. There emerges from their bellies a drink, varying in colors, in which there is healing for people. Indeed, in that is a sign for people who give thought or mediate. Okay, so in this verse, Allah is talking about the importance of honey. Honey, which we get from the bellies of the honeybees, it has a very good healing property. Okay, so we all know that eating honey is shifa according to Quran and Sunnah. So whichever ailment you have, whether you're sick, you have any type of a medical condition, or you just have a cut or a bruise, immediately we apply honey on it, or we need to eat honey so that we get quick healing, inshallah. Okay, so this has been stated in the Quran, and it is a fact that honey, it has a healing property in it. And in fact, it is coming from the bellies of the honeybees, and it has an enzyme in it which helps in healing faster or quicker. Okay, so if we are strictly following the Quran and Sunnah, there are many foods in fact mentioned in the Quran, not only the honey, but also about the black seed, about cinnamon, about dates, and many other fruits and vegetables. If we eat, in fact, we will get benefit from it, inshallah. All right, so that is about this verse. Let's talk about the objectives for this topic. Would anyone want to read out the objectives for this topic? Who wants to read them out? Girls? Yeah. Can you please read out the objectives from here?
Yes. In this chapter, you will find out about enzymes and what they do, how enzymes are affected by the temperature and pH, why enzymes are affected by the temperature and pH, to investigate the effects of temperature and the pH on enzyme activity, how to plan, carry out, and evaluate your own experiments on enzyme activity.
Thank you so much. So these are the objectives for this topic which you are going to learn further. All right. And so let's start by doing a KWL. I believe we already studied, I introduced you to enzymes. We were discussing about it in our chapter 4, right? As to enzymes, how they function and how they work. Do you remember?
Yes, Miss.
Yeah, so what can you interpret by looking at this picture on the screen? What is it about? What is it explaining about? Why only 9 attendees? Where are the rest? Okay, 14, alhamdulillah. Yes, ladies, what do you understand by this picture? It's written on it as well. All right, you can see the blue part. This is the enzyme, and it works well when it comes into contact with the substrate. We discussed about this, about the lock and key mechanism. As to the enzymes, they function well only when they come into contact with the right substrate. Right? All the enzymes, they cannot function for any metabolic reaction. Okay, a specific enzyme can work only for a specific metabolic reaction. So the enzymes, of course, they act as catalysts in order to speed up the reactions, but for the enzymes to function properly, they need to come into contact with the right substrate. Okay, so as you can see in this picture over here, these are the A and B molecules. When they fit into the enzyme like this, the enzyme can work at its best. All right, so this is what is being explained here. We are going to talk about in this topic further as to how the enzymes work. All right. So let's also talk about the objectives as to what you learn for the core and also the supplement papers of your IGCSE. Can someone please read out the core and the supplement objectives from the screen?
Miss, can I?
Yes, sweetie.
Okay, enzymes core. Number 1, define enzymes as proteins that function as a biological catalyst. 2, and supplement, explain enzyme action with reference to the complementary shape of the active site of an enzyme and its substrate and the formation of a product. Investigate and describe the effect of changes in temperature and pH on enzyme activity. Explain the effect of changes in temperature on enzyme activity in terms of kinetic energy, shape and fit, frequency of effective collisions and denaturation. Explain the effect of changes in pH on enzyme activity in terms of shape and fit and denaturation.
Thank you so much, sweetie. So this is what you're going to learn, and you have to be aware of by the end of this topic. You need to know all these questions, whether you're answering a core paper or a supplement paper in your IGCSE. All right, ladies. So let's start by watching a video on enzymes. It is a very interesting song. Of course, it is a song, but it gives you an insight as to what are the enzymes and how they work. It is quite interesting. So let's go ahead and watch it. Is the audio clear? Is the audio clear, ladies?
Yes.
Oh, okay, great.
Enzymes, gotta catalyze reactions. Active sites specifically shaped for the substrate. Enzymes made of amino acids. Gotta change shape slightly to accommodate the substrate. Helicase, DNA polymerase. Fun, make an enzyme substrate complex. Enzymes, gotta catalyze reactions. Active sites specifically shaped for the substrate. Enzymes made of amino acids. Gotta change shape slightly to accommodate the substrate. Helicase, DNA polymerase. Fun, make an enzyme substrate complex. Enzymes, gotta catalyze reactions. Active sites specifically shaped for the substrate. Enzymes made of amino acids. Gotta change shape slightly to accommodate the substrate. Helicase, DNA polymerase. Fun, make an enzyme substrate complex.
Okay, so that was an interesting way of learning about enzymes and its functions, isn't it? Did you all enjoy it?
Yes, miss.
Thank you so much. So let's discuss about enzymes and what is their role and why are they known as biological catalysts. Okay, but before that, on your page number 49, you have a quick introduction about enzymes. Okay, so you can see about enzymes explained from the forensic point of view. Have you ever heard of the forensic science or the forensic laboratories and forensic scientists? Have you ever heard of these terms, ladies?
Yes, Miss.
No.
Yeah, where did you hear about it?
In crime related stuff.
Yes, very good. So wherever there is a crime, they investigate about the various things, about the evidences as to how the crime has taken place, and they take various evidences from fingerprints to the various things they use, and also a glass from where they must have drunk, or the criminal must have used a cigarette, or must have drank a can of Coke or Pepsi and just thrown it there. Okay, so many things are taken from that place as evidence in order to investigate as to who the criminal is or who actually did the crime. Okay, so such type of scientists who investigate about the crime are known as forensic scientists, and the science itself is known as forensic science. Okay, so forensic science is the use of scientific techniques to obtain evidence relating to the crimes. So why are we talking about forensic science under the topic enzymes? Let's check out. Now the human saliva, it contains an enzyme called salivary amylase that helps to digest starch in the mouth. Okay, so the digestion starts from the mouth itself, especially the digestion of the carbohydrates like the starch. And the enzyme which starts the digestion of the starch in the mouth is known as salivary amylase, which is present in our saliva. All right. Okay, so forensic scientists can test surfaces for the presence of human salivary amylase. Okay, what do? They check for any glass or a cup which the criminal must have used, as I told you, or maybe a cigarette, or maybe a bottle of soft drink or wine or coke or anything. So this evidence they check in order to to find out the enzyme present, which is salivary amylase, which is present in the saliva. Okay, so by investigating the DNA present in the salivary amylase, this DNA can be matched with of course, the persons on which there is a doubt, or the susceptible, whoever is suspicious. So they check the DNA of that particular person, and they try to match it with the DNA present in the salivary amylase of the saliva on the various surfaces which have been used. Okay, so this can help to determine whether a person was present at the crime scene or no. Okay, so when the test first came in, it was only able to detect the activity of amylase, that is, whether starch was digested. Although this could be useful, a positive result, it did not prove that a person had left saliva at the scene. This is because amylase is also produced by many other organisms such as bacteria and fungi. All right, so this amylase is not only produced by humans, it is also produced by gorillas and rats and bacteria and fungi. So earlier it was not known, and this particular enzyme was not helpful a lot in the forensic science. But later, in the late 1980s, a new test that could detect human amylase directly was introduced. Okay, so it was very important for them to check if the salivary amylase was from humans or from gorillas or from bacteria. Why? Because again, of course, there will not be any gorillas in the crime scene, but they can be rats, they can be bacteria, so they can also be any type of fungi who must have released this salivary amylase, isn't it? However, this test can still give positive results for amylase from other organisms, including rats and gorillas. So although it is very unlikely that a gorilla was present at the scene of the crime, it is often possible that a rat might have left the saliva behind, isn't it? So today, forensic scientists are also able to search for check cells within a saliva sample, and if they can find any, then they can test the DNA in them, and this can then provide evidence that a person, it can link it to a particular person at the crime scene. So we are talking about all this here because of this enzyme, salivary amylase, and how this enzyme can lead the forensic scientist to the criminal. All right. So let's talk about enzymes now. What are enzymes? So let's start with chapter 5.1, page number 50, about biological catalysts. Okay, so enzymes, they have been given a name as biological catalysts. Why? Because many chemical reactions can be speeded up by substances called catalysts. Okay, so these enzymes, they help in speeding up the reaction, though they will not undergo any change themselves, but they help in speeding up this reaction. For example, if you're talking about the pepsin, pepsin is an enzyme, a digestive enzyme, which acts on the food, especially the proteins in the stomach. Okay, so this breaking down of proteins in the stomach becomes faster. This particular reaction, it is speeded up when pepsin is released. Okay, so this pepsin enzyme, it helps in speeding up the reaction, though it will not undergo any change by itself. That is why the name catalyst has been given to the enzymes. So a catalyst, it alters the rate of a chemical reaction without being changed itself. So within any living organism, the chemical reactions that take place all the time, isn't it? We studied this in our previous topic, that many metabolic reactions, they keep on taking place in our body simultaneously. Can you give me examples of the the metabolic reactions that might be going on in your body now while you're sitting and attending this class? Ladies?
Respiration and digestion.
Very good. And? Yes, the respiration, we are respiring, whether we are sitting and talking or sleeping, and the digestion process is taking place. And if you're drinking water, that is getting digested, and also the the urine is getting ready for excretion, meaning the excretion process is also taking place. And there are other metabolic reactions, meaning the reactions taking place when our brain is sending signals to our various organs. Maybe you're writing down something, and again the brain is sending a signal, isn't it? All right, so there are many metabolic reactions which are taking place simultaneously in your body, and almost every metabolic reaction is controlled by catalysts called enzymes. Okay, so without enzymes, the reactions would take place very slowly or not at all. Okay, so enzymes play a very important role in these metabolic reactions to take place, be it in plants or animals. The enzymes, they not only helping in speeding up the reaction, but they make the reactions happen. All right, without enzymes, the reactions would take place very slowly or not at all. Okay, so enzymes ensure that the rates of metabolic reactions are great enough to sustain life. All right, so you might have to memorize these key definitions of a catalyst and enzymes. Can someone please read out the definitions?
Miss, can I read?
Yes, sweetie.
Catalyst, a substance that increases the rate of a chemical reaction and is not changed by the reaction. Enzymes, proteins that function as biological catalysts.
Very good. Thank you. So these are the definitions you might have to memorize. You also have these definitions in your textbook on page number 50. Now let's talk about enzymes as to how they help in the chemical reactions. Okay, so we already discussed that the sum of all chemical reactions going on within a living organism is known as metabolism. Metabolism is a sum of all the reactions taking place together. So metabolic reactions can again be categorized into anabolic reactions and metabolic reactions under metabolism. Okay, so anabolic reactions, they build up large molecules from smaller ones and usually require an input of energy. Okay, so in these reactions, the smaller molecules, they come together to form larger molecules. For example, as we studied under chapter 4, carbohydrates, we studied the classification of carbohydrates into monosaccharides, disaccharides, and polysaccharides, isn't it? And we also studied that a polysaccharide molecule, be it glycogen or starch or cellulose, all of these, they are made up of long chains of glucose units or monosaccharides which are linked together, right? So this reaction which you can see over here, the condensation of the glucose molecules into the polysaccharide glycogen is an example of anabolism or anabolic reaction. Okay, so in this reaction you can see a single glucose molecules like this, they come together and they condense to form long chains of a glycogen molecule. We know the glycogen, it is a polysaccharide, of course, and its function in the body, what is the function of glycogen in the body, ladies?
Stored energy.
Very good. Glycogen reserves are stored in our liver and muscles as reserve energy so that we can use it whenever we need it in excess. Okay, so whenever we need extra energy, these glycogen reserves are broken down and converted to glucose so that we get immediate energy. Alright, so this condensation of glucose molecules into the glycogen polysaccharide, it is an example of an anabolic reaction, wherein the smaller molecules, they combine together to form larger molecules, and it usually requires an input of energy for these reactions to take place. Okay, so this is an example of how an anabolic reaction might take place. Okay, so this is how the enzymes, they help in the anabolic reactions by coming into contact with these A and B molecules together. Okay, so we are going to talk about how these enzymes help in the anabolic reactions further now. Alright, now what are catabolic reactions? So anabolic is, we studied that making of larger molecules from smaller molecules in which energy is required, right? And catabolic reactions are exactly opposite to that. Catabolic reactions, they break down the larger molecules into smaller ones and often release energy, all right? The anabolic reactions, they require energy, and catabolic reactions, they release energy. Similarly, anabolic reactions, they are the reactions in which the smaller molecules are joined together or condensed together to make larger molecules, whereas in catabolic reactions, the larger molecules, they are broken down into smaller ones and often energy is released. Okay, so one of the examples of a catabolic reaction is the breakdown of glucose into carbon dioxide and water by respiration. Okay, so what happens during the process of respiration? We discussed about this in our previous topics as well. So whenever we breathe in oxygen, when oxygen comes into contact with glucose plus oxygen, it gives rise to carbon dioxide and water, and energy is released during this process. And this equation is how the respiration is explained, isn't it? So this particular reaction wherein the larger molecules of glucose, they are broken down into smaller molecules of carbon dioxide and water is an example of an catabolic reaction. Okay, ladies, so is it clear about the anabolic and catabolic reactions? Any questions so far?
So, Miss, when the catabolic reaction will occur?
When we respire. There are many examples of catabolic reactions apart from respiration in our body. But one of the examples, in order to explain you clearly, is when the respiration takes place. When you respire, what happens when you breathe in oxygen? Yes, sweetie. The oxygen, it comes into contact with the glucose in the food which you eat, okay, and the glucose is broken down, and this is how the energy is released, the energy which you get in order to carry out your daily activities, for talking, for walking, for doing various sports, exercise. You get this energy by the process of respiration. Clear? So this is an catabolic reaction. Clear, girls? Any questions?
Yes, it's clear.
Okay. So this is how the catabolic reaction takes place. It is explained with a picture. Let's also discuss as to how it is explained in your textbook. Okay. So you may look at page number 50 from your textbook. All right. Now let's discuss about the information we have in your textbook on page number 50. We already discussed about why enzymes are known as catalysts, and we also discussed about the definitions of catalyst and an enzyme, right? So let's move on. For example, inside the alimentary canal, large molecules are broken down to smaller ones in the process of digestion. Right? So in the process of digestion, what happens in your alimentary canal? The larger molecules, they are broken down into smaller molecules. So will this be an example of an anabolic reaction or a catabolic reaction?
Catabolic.
Very good. So it is a catabolic reaction. So these reactions are speeded up by enzymes. So enzymes, they play a major role in speeding up either an anabolic reaction or a catabolic reaction. For example, a different enzyme is needed for each kind of food. For example, starch is digested to the sugar maltose by an enzyme called amylase, and protein is digested to amino acids by protease. Like this, there is a different enzyme which works on different kinds of foods. Okay, in the foods which we take, as we discussed in our chapter 4, we take proteins, we take carbohydrates, we take lipids. right? So the carbohydrates which we take in our food, they are digested by the enzymes known as amylase, be it salivary amylase or pancreatic amylase. So all the names of the enzymes which digest the carbohydrates, they are given the name amylase. Okay, be it the enzyme present in our saliva, be it the enzyme present in our pancreas or various parts of our digestive system. Similarly, the name of the enzyme which helps in breaking down proteins, it is known as protease. Okay, and similarly, the names of the enzymes which help in breaking down the fats, they are given the names like kinase. So whenever we are going to talk about in detail about the various enzymes which help in the process of digestion and various other metabolic reactions which take place in our body. So these enzymes are not only present in animals, they are also found in plants. For example, in germinating seeds where they digest the food stores for the growing seedling, many seeds they contain stores of starch. We already studied that even in plants, the excess of glucose, it is stored in the form of starch in the plants, right? And whenever the plants needs extra energy, they can easily convert the starch into glucose, right? So as the seed soaks up water, the amylase is activated and breaks down the starch to maltose. And the maltose is soluble and is transported to the embryo in the seed. The embryo uses it to provide energy for growth and also to provide glucose molecules that can be strung together to make cellulose molecules. So this is the energy which is required by the seed for it to germinate and produce a plant. Right? From where does the seedling get this energy? We studied that from the endosperm it gets the energy, and the endosperm is the coating of the leaf, and wherein the starch it is converted to maltose, and maltose can be converted to glucose. So another enzyme which speeds up the breakdown of the substance is known as catalase. Okay, so catalase works inside the cells of living organisms, both animals and plants. For example, in liver cells or potato cells. It breaks down hydrogen peroxide to water and oxygen. Okay, so this is another example of an enzyme which speeds up various metabolic reactions both in plants and animals. It is known as the catalase enzyme. And where does it work? It works inside the cells of living organisms in both plants and animals, in the liver cells in animals, and in potato cells in the plants. So what does it do? It breaks down the hydrogen peroxide to water and oxygen. So this is necessary because hydrogen peroxide is produced by many of the chemical reactions which take place inside the cells. Okay, so hydrogen peroxide which is produced as you can say as a byproduct of various metabolic reactions. This hydrogen peroxide is broken down further with the help of this enzyme which is known as catalase. Alright, so I'm simultaneously explaining what is present in your textbook as well so that we don't miss out on any information, ladies. So after this, I will come back to this anabolic and catabolic reactions which are in our presentation. Alright, so this is about the catalase. So this is necessary because hydrogen peroxide is produced by the many of the chemical reactions, and hydrogen peroxide is a very dangerous substance and must be broken down immediately. For this hydrogen peroxide to be broken down, this enzyme catalase, it is very, very important. So not all enzymes help to break things down. Many enzymes help to make larger molecules from smaller ones. So all these reactions which we studied so far about breaking down the larger molecules into smaller ones are all examples of catabolic reactions, right? We spoke about the breaking down of food in the alimentary canal, and we also spoke about the breaking down of hydrogen peroxide into simpler molecules. So all these would be examples of catabolic reactions. Okay, so enzymes, they not only help in catabolic reactions, they also help in the anabolic reactions, wherein the small molecules, they are joined together to form larger molecules. Okay, so one example of this kind of enzyme is starch phosphorylase, which builds starch molecules from glucose molecules inside the plant cells. All right, so we are going to discuss about the various enzymes which help in various reactions in this topic further, inshallah. Any questions so far, girls? Is it clear? All clear? Any questions?
Yes, it's clear.
All right, so we are going to discuss further about this topic in our next class, inshallah. No homework so far. I want you to complete the homework for the last lesson first and also to go through the topic which we covered today. All right, so you have a great day ahead. Let's meet in the next class, inshallah. Take care.